Hello everyone. Let's continue our discussion on protein synthesis. The second part is on prokaryotic translation. Now, the overall process of translation in both prokaryotes and eukaryotes that can be described in three stages. That includes initiation, elongation, and termination of translation. In the first session, we discussed the various requirements for protein synthesis or translation process, including the different components like the charged tRNA, the synthesis of the charged tRNA, the structure of the ribosome, and even the structure of the mRNA that is used in the translation process. All these were briefly discussed in the previous session. So, once all these things were ready, there is the processed RNA, which is the storehouse of the information for translation, and the charged tRNA with the corresponding amino acids, and the ribosome or the protein synthesis machinery, all these things were available readily. Then we can proceed with the synthesis of proteins. That is a translation process. In the process of translation, as I mentioned, the first stage is the initiation of the translation. In the initiation of translation, a translational complex forms, and the RNA brings the first amino acid into the polypeptide chain to bind and to start the translation of the codons on the mRNA. Then the second stage, that is the elongation stage, in which the tRNA brings more amino acids and add to the growing polypeptide. In the termination process, some release factors recognizes the specific stop codons or one among the three stop codons that is available, and the translation complex dissociates, releasing the polypeptide and terminating the translation process. So, in this session, we will discuss these three stages of the translation, especially the prokaryotic point of view of translation, in detail. This is another representation, schematic representation of the prokaryotic translation, in which the different components of the translation, including the charged tRNA, the two subunits, that is in the prokaryotic system, we have the 30S and 50S subunits of the ribosome is represented here. The mRNA with this 5' and 3' ends, and the AUG, that is the start codon, then the stop codon, that is UAG, and in between there is a translated regions or the coding regions of the mRNA, and all regions of the mRNA were represented in that slide. Now, in the first stage of the translation, that is the initiation stage, the initial mRNA, the first tRNA carrying the first amino acid need to occupy the first start codon, that is initiation codon, by the translational assembly. That translational assembly includes the mRNA, the small and large subunits of the ribosome, and the tRNA, that is a charged tRNA, correctly positioned at the start codon, that is AUG codon. So then that is the overall process of the initiation. Then in the elongation stage, as I mentioned, the second site, that is the A site of the amino acid is, the ribosome is occupied by the second tRNA that is carrying the second amino acid, and then that peptide or the existing amino acid in the P site will be transferred to the amino acid in the A site, and that chain will grow by continuous addition of the incoming amino acids that is added to the A site. Then in the third stage, as I mentioned, the termination occurs by the binding of the release factors to the stop codon, and subsequent release of the completed peptide and the translational assembly, making it available for further recycling of these translation assembly components for further translation process. Now, in the first stage, that is initiation stage, we have various initiation factors that is involved. In prokaryotic system, we have three initiation factors, that is IF1, IF2 and IF3. Then the second stage, there are other items that is involved, like the first amino acid that is incorporated, that is a formyl methionine in case of prokaryotes. Note that the eukaryotic initiation is done by incorporation of normal methionine by recognizing the initial AUG codon. But in prokaryotic system, always the first amino acid is incorporated is not methionine but a formyl methionine with a formyl group. And it is recognizing the same AUG codon, which is coding for the methionine, but initiation process, which recognizes the first AUG codon in the mRNA, is actually that is done during the initiation process, and at that position only we are using this formyl methionine. And subsequent AUG codons in the prokaryotic mRNAs were all incorporated with the normal methionine, not formyl methionine. So only at one position, the first AUG codon, which is the initiation codon, only that formyl methionine is used for a prokaryotic initiation. And then there are ribosome binding sites, specific ribosome binding sites, which is present in the mRNA, which is upstream of the start codon, and they are known as the Shine-Delgarno sequence, and which is having a specific sequence of AGG, which is having some complementary sequence to the ribosomal RNA proteins. The ribosomal RNAs. So that complementary base pairing between the ribosomal RNAs and the Shine-Delgarno sequence of the mRNA will correctly position the mRNA with the AUG codon at the P site of the ribosome assembly. Okay, so the correct positioning of the mRNA by positioning the first initial these are correct positioning of the mRNA or the binding of the mRNA to the ribosomal assembly is done by the specific base pairing of the Shine-Delgarno sequence with the ribosomal RNA sequences. Okay. Now the energy from GTP is used for, actually it is bound, this GTP is bound to the IF2 or initiation factor 2, and that energy of the bound GTP of the IF2 is used to complete that assembly formation. Now, here we have the 30th subunit of the small subunit of the ribosome is here. 3 initiation factors and GTP, the requirements all are here. And the binding of the 30th subunit by initiation factors actually prevents this ribosomal subunit, small subunit, to reassociate with the larger subunit. Normally, the larger subunit and smaller subunit of the ribosomes will associate and dissociate. This process goes on. But while it is or the initiation factor 3, especially the initiation factor 3, when it is bound to the subunit, 30th subunit, it prevents the further reassociation of these smaller subunit with the larger subunit, keeping it or making it available for binding of the other factors and the Shine-Delgarno sequence of the mRNA. So this initiation complex, initiation process is done by initially binding of the initiation factors, which keeps that ribosomal subunit, small subunit as such, instead preventing its reassociation with the larger subunit, and then the remaining factors like remaining items like the mRNA and other initiation factors will, or even tRNA, the charged tRNA will bind to the initiation complex. So in the initiation process of the protein synthesis, the first codon translated in all mRNAs is the start codon or the initiation codon, that's the AUG codon, which is actually coding for methionine. Two different tRNAs are used for two types of AUG codons. tRNA methionine is used for initiation codon and is called initiation tRNA, whereas tRNA FMET is tRNA methionine is used for internal AUG codons. That means tRNA FMET or formyl methionine. FMET means formyl methionine and methionine tRNA or meth. So the tRNA, there are two different specific types of tRNAs are used for initiation and elongation process in prokaryotes. tRNA with formyl methionine is recognizing the initiation AUG codon, and subsequent AUG codons in prokaryotes were recognized by tRNA methionine or methionine charged tRNAs. And so we have a prokaryotic system in which the AUG codons were recognized by two different types of tRNAs. The initiation AUG codon recognized by formyl methionine tRNA and the subsequent AUG codons or internal AUG codons in the mRNA were recognized by methionine tRNA. And these tRNAs were specifically synthesized by formyl methionine tRNA synthetase and methionine tRNA synthetase. So we discussed it in the previous session. In prokaryotes, the first amino acid of a new protein is the normal or N-formyl methionine, that is abbreviated as FMET, and hence the amino acyl tRNA used in initiation process is FMET tRNA FMET or formyl methionine tRNA charged with formyl methionine. A short sequence rich in purines, that is AGG, AGGU. So that is a particular sequence known as a Shine-Delgado sequence, which lies towards the 5' end of the initiation codon and all of the mRNA, and that is actually complementary to some of the parts of the 16S rRNA, which is present in the small subunit, small ribosomal subunit. So the specific complementary base pairing of the Shine-Delgado sequence and the 16S rRNA sequence enables the correct positioning of the mRNA in the 30S subunit. Therefore, this is the binding site for this Shine-Delgado sequence is the binding site for the 30S ribosomal subunit, which then migrates to the 3' direction or towards the right side of the mRNA. So this 30S ribosomal assembly, once it is bound to the Shine-Delgado sequence, then it migrates or moves towards the 3' direction of the mRNA until it finds the first AUG codon or AUG initiation codon. Once it is located and correctly positioned at the P site of the ribosome, then the tRNA, formyl methionine tRNA, charged formyl methionine tRNA will occupy that position. Or that if initiation codon, which is correctly positioned by scanning downstream to the correct position of the first initiation codon, and then the tRNA, which is charged with formyl methionine, will be binding to that region. The initiation of protein synthesis requires proteins called initiation factors or IFs, and in prokaryotes, 3 initiation factors, that is IF1, IF2 and IF3 are essential. Because of the complexity of this process, the exact order of binding of these 3 different factors and formyl methionine tRNA is controversial. Which one binds first? There are different opinions about that one. So this slide shows the different initiation factors for both prokaryotic and eukaryotic system, and which is used for initiation process. And in prokaryotic system, we can see that this IF1 is the first factor which prevents the premature binding of the tRNAs to the A site. So without IF1, the specific binding of the tRNA to the A site is not possible. IF2 is the factor that facilitates the formyl methionine tRNA or the binding of this formyl methionine tRNA to the 30S subunit. So the initiator tRNA, that is the formyl methionine tRNA, is specifically bound to the P site of the 30S subunit by the help of IF2. Now the IF3, it binds to the 30S subunit and prevents the reassociation with the 50S. So this enhances the specificity of the P site for the formalin methionine tRNA binding. So the initially IF3 is critical because IF3 keeps the 30th subunit free from binding to the 50th subunit, and that facilitates the binding of the FMET tRNA and other factors or other initiation factors to the complex. So IF3 binding keeps the 30th subunit standalone without reassociation with the 50th subunit. So this picture shows the 70th subunit with associated 30th and 50th units. Then normally there is a dissociation and reassociation of these small and large subunits. Now this is actually facilitated by IF1 also. Now once the IF3 binds to the small subunit, then the larger subunit cannot bind, keeping the small subunit free for binding of the other factors. So then you can see that the GTP bound IF2 and IF1 all will bind to the smaller subunit. Then the mRNA and the formalin methionine tRNA, which is the initiator tRNA, will come and bind to the initiation complex. Then that 30th initiation complex is formed now with all the three factors and GTP and formalin methionine and mRNA. So that is the 30th initiation complex. And once it is formed, the IF1 and IF3 will be released. So once the IF3 is released, the inhibition for binding of 50th subunit will be relieved. So the 50th subunit can again come and bind, complete the process of the complete formation of the 70th subunit, 70th ribosomal complex, which is required for translation. So the initiation is now over by releasing of the GTP and IF2, and PPI is also released. So this entire complex is formed, and after release of the initiation factors, now the mRNA is correctly positioned in the ribosomal assembly, and that is the 70th initiation complex. So this is another representation in which IF3 is bound and IF1 is bound to the 30th subunits, and then mRNA is bound, then tRNA is bound, and this is the 30th initiation complex. Then other factors IF1, IF2, IF3 releases, and the larger subunit associates. This is the 70th ribosomal initiation complex. This is another representation, and it is working in the same manner: reassociation and association and reassociation of larger and smaller subunits. The mRNA is here. Different factors will bind to the smaller subunit. mRNA and factors, that order may change. For example, sometimes someone will say that initially mRNA binds, then tRNA binds. That may vary. In both ways it can occur. Initially the tRNA can occupy the space, and then the mRNA can come and bind, or the initially mRNA can bind and later the tRNA can bind. So in both ways it will work. Anyway, the pre-initiation complex of 30S pre-initiation complex is formed in both ways, by binding of the tRNA or rRNA, whatever initially. Then after release of the remaining factors, or IF1 and IF3, then the 50S binds and completing the formation of the 70S subunit, 70S complex or 70S initiation complex, subsequently leading to the elongation. And for the elongation, we require binding of elongation factor TU, and we will see that later. So that remaining steps in the elongation and translocation will be discussed later. So this is another representation. Okay. So in the summary of initiation, we can see that the dissociation of the 70th subunit into 50th and 30th subunits under the influence of IF1 or initiation factor 1. So the dissociation is facilitated by initiation factor 1. Binding of the IF3 to the 30th subunit prevents the reassociation and keeping that smaller subunit available for binding of the further factors. Binding of IF1, IF2 and GTP alongside GTP is the next step, which contributes to the 30th pre-initiation complex. Okay. Then also the mRNA and formalin methionine tRNA will bind to the 30th initiation complex, and that is actually pre-initiation complex, 30th pre-initiation complex. Okay. And these two components, that is the mRNA and formalin methionine tRNA, are components that can apparently bind in either order. That is, anything can bind first. There is no specific order mentioned. But the IF2 sponsors, or initiation factor 2 sponsors the tRNA binding, and IF3 sponsors mRNA binding. So these factors is also having specific roles. tRNA binding is promoted by IF2 and mRNA binding is promoted by IF3. In each case, the other initiation factors also help in overall complex formation. Now, binding of the 50S subunit with the loss of the IF1 and IF3, IF1 and IF3 is released, which enables the 50S subunit binding and completing the 70S complex formation, or the complete ribosome assembly is formed by release of the IF1 and IF3. Dissociation of IF2 from the complex or with the utilization of the GTP, with simultaneous hydrolysis of the GTP, results in the complete production of a or 70S initiation complex. Now it is ready for elongation. In the elongation stage, there we need the binding of the second tRNA carrying the second amino acid. Then the peptide bond should be formed between the first amino acid or formyl methionine and the next incoming amino acid. Then there needs to be a translocation process which requires the movement of this mRNA through the ribosomal assembly. In the this elongation stage, actually the first step of the elongation stage is occurring at the P site, contains either methionine or as you know, in case of prokaryotic system, it is formyl methionine, or eukaryotes, it may be methionine. And so the P site of the ribosome is actually occupied by methionine or formyl methionine. But prokaryotes, since we are discussing prokaryotes, that is formyl methionine. So that methionine tRNA or formyl methionine tRNA is occupied at the P site initially. P site is the central position of the assembly. E site is the exit site, P site is central, and A site is towards the 3' end of the mRNA. A new aminoacyl tRNA or the second tRNA carrying the amino acid or charged tRNA arrives and bound to the EFTU. EFTU is a factor which is known as a temperature unstable factor, elongation factor which is temperature unstable, temperature labile. So that is a EFTU. So this elongation factor TU bound with the two GTP molecules that will come and bind to the initial, the second tRNA, aminoacyl tRNA. So here you can see that this is the initiation complex or 70S initiation complex, and elongation factor TU, which will bind to the second charged tRNA, will form a complex, and then that will occupy the A site or aminoacyl site of the assembly. Then if the codon anticodon matches, so if there is a specific pairing of this codon and anticodon, hydrolysis of the GTP is occurring, and that GTP or that energy is used to insert a new aminoacyl tRNA into the A site. So once these matches, if it is a correct correct codon anticodon pairing, then GTP is hydrolyzed, and that GTP, if it is hydrolyzed, then EFTU complex, GDP complex is released, and that aminoacyl tRNA will be correctly positioned at the P site, sorry, A site. Now, the polypeptide chain is transferred from the the P site to the A site. So the second stage, this is the P site or initial amino acid, and in prokaryote that is a formyl methionine. So that amino acid at the P site will be transferred to the amino acid at the A site. So that reaction is known as peptidyl transferase reaction. So this peptidyl transferase is a ribozyme, that is a RNA with enzymatic activity. So which is a part of the ribosome, which is not an external enzyme. It is a part of the RNA that is present in the ribosome assembly. So that is having that ribosomal structural component or an RNA which is present in the ribosome structure, which is having an enzymatic activity. So that is the ribozyme. So this peptidyl transferase activity is an activity of the RNA protein, rRNA which is present in the structure of the ribosome or structural component of the ribosome. So that peptidyl transferase is a ribozyme. One of the rRNAs act as an enzyme to catalyze the transfer. Then the energy for this peptide bond formation is provided by the ester bond between the amino acid and the tRNA. So this amino acid and tRNA, this is connected. There is an ester bond between the amino acid and tRNA. So that ester bond is transferred, and that is transferred to the amino acid at the A site. So that peptide transfer reaction, in effect, it transfers the amino acid to the incoming amino acid. Then there is another factor known as EFG, which binds to with one GTP, and that EFG it is the hydrolysis of this GTP, which is bound with the EFG. It causes a shift of the ribosome along the mRNA until the growing chain is back in the P site, and the empty RNA is in the E site. So the next one is the EFG is another factor. So EFG, elongation factor G, bound to the GTP, will act on this one, and it slides the mRNA in one direction, so that the contents of the A site will be moved to the P site. Contents of the P site will be moved to the exit site. So the RNA, the ribosomal assembly will slide through the mRNA, rearranging the position of the contents of the A will be moved to the P. Contents of the RNA, the tRNA at the P site will be moved to the E site. And that process is actually the process of a translocation. So the peptidyl transferase is the first step, and second step is a translocation, and that is actually with the aid of a EFG and a GTP complex. So that's the next step, or the third step in translation is termination, and the termination is occurring when a stop codon is encountered. As we discussed, we have three different stop codons which is present, and a release factor may bind to the stop codon. We have two types of release factors in prokaryotic system. One is RF1 and the second one is RF2, and there is release factor 1 will recognize stop codons UAA and UAG, whereas release factor 2 can recognize UGA and UAA. So total we have three stop codons, and it will be recognized either by RF1 or RF2. So that release factor will bind whenever that release factor is present at the A site. If there is an A site with a stop codon, then there will not be any corresponding anticodons, or there will not be any corresponding tRNAs for that stop codon. So that position is occupied by the release factor, and the release factor may be RF1 or RF2. The peptide chain transfers to a water molecule instead to an amino acid, creating a free hydroxyl group. So a free hydroxyl group is created. That is, peptide is now from the end of the tRNA, the peptide is released by transferring this bond to a water molecule and releasing the peptide. Now the tRNA is free. The post-translational complex falls apart. So after this, the translational complex falls apart. The translational complex at that time includes smaller and larger subunits, mRNA, RNA, tRNA, and the release factors. All were part of the translational complex at that time, and all fall apart, or they will be dissociated, and it will be made available for recycling and reuse. So this is another illustration of prokaryotic translation, and here you can see that there is a translation bubble in which the DNA duplex is unwound, and here is the transcription process that is occurring. As we discussed, since it is a transcription and translation, co-transcription and co-translation process is occurring, so that is it's a simultaneous transcription and translation. So when a free 5' end of the RNA is made available, this ribosome assembly starts binding at the end, and it starts translation. And in prokaryotic system, as I mentioned, this is a rapid process with or a simultaneous process of transcription and translation that is occurring. So this is another illustration of the overall process of a gene expression in a prokaryotic system. So the transcription and translation simultaneously happening in the cytoplasm. Okay.